school, I don't have access to YouTube. All right, so let's get started. We are doing the topic process of meiosis, which is chapter 6.2. And let me tell you the page number which we are going to do today. Okay, so we are going to do page number 164 and 165. So please open page number 164 and 65 from your textbook, ladies. So let's start. Bismillah ar-Rahman ar-Rahim. All increase as a beneficial knowledge. The objective for the topic would be to compare and contrast the two rounds of division in meiosis and to describe how haploid cells develop into mature gametes. Okay. Under the KWL, let me ask you questions from the content I covered yesterday. The key concept for this topic is during meiosis, diploid cells undergo two cell divisions that result in haploid cells. So unlike the process of mitosis, the process of meiosis happens in two stages, which is known as Meiosis I and Meiosis II, which are the two rounds of the division in meiosis. Right? Okay, so let me also display the textbook page here. Give me a moment. All right. So yesterday we were discussing as to how the cells go through two rounds of division in meiosis. Can someone tell me what are the four stages of those two rounds? What are the four stages which happen in Meiosis I? Lana, go ahead.
The prophase, the metaphase, the anaphase, and the telophase.
Perfect. So these are the four stages: PMAT, prophase, metaphase, anaphase, and telophase. And the four stages which happen in Meiosis I, they are known as Prophase I, Metaphase I, etc. And the stages which happen in Meiosis round 2 are named as Prophase II, Metaphase II, etc. Leen, could you please tell me what is the difference between homologous chromosomes and sister chromatids?
Okay, so homologous chromosomes are two pairs of chromosomes, one from the mother and one from the father, that are very similar in terms of general appearance. The sister chromatids are like the two parts of each of the chromosomes. Each of these chromosomes contains two sister chromatids, which are joined by the centromere.
Perfect. All right, very good. So that is the difference, thank you so much, Leen, between homologous chromosomes and sister chromatids. Now, homologous chromosomes are divided in Meiosis I, whereas sister chromatids are divided in Meiosis II, right? So yesterday we were going through these four stages, day before yesterday, I believe, which are the four stages which happen even in mitosis and meiosis both. Okay, so yeah, here you can see the four stages in Meiosis I. It divides the homologous chromosomes further. So did you watch the video about centrosomes and centrioles, Leen? Did you understand now what are centrosomes and centrioles?
Yes, I'm sorry I couldn't watch the video because I had lessons when you sent it.
All right, no problem. You may watch it later. So now, do you know what are centrosomes and centrioles?
Yes, they're in this picture. There are the blue things that go to opposite sides in order to divide the chromosomes.
Yes, perfect. So basically, they are also present in the nucleus of the cell, and they also help in the cell division. All right. Can someone tell me the various things which happen in Prophase I, the sequence of the events which happen, which leads how it happens, and how does the division of the nucleus take place in Prophase I, Metaphase I, Anaphase I, and Telophase I, starting with Prophase I. Is it clear over here, or do you want me to display the PPT? Yeah. Who wants to do it? Or do you want me to explain it once again if you have any doubts or questions so that they might be cleared? All right, so let me do it again. So now we know the differences between sister chromatids and homologous chromosomes, isn't it? So meiosis 1 and 2 each have 4 phases, which are similar in mitosis as well. And we'll talk about how the division of one cell takes place in meiosis 1. So meiosis 1 occurs after the DNA has been replicated, and it divides the homologous chromosomes in 4 phases. As we discussed in meiosis 1, the homologous chromosomes are divided further, whereas in meiosis 2, the sister chromatids are divided. So we'll talk about the division of the homologous chromosomes first. So as you can see in this picture, you can see these homologous chromosomes, 2 pairs in the nucleus, right? 2 from the mother and 2 from the father. Now, in prophase 1, the nuclear membrane breaks down. This membrane which surrounds the nucleus, it breaks down. And the centrosomes and centrioles, they begin to move, which are also present in the nucleus. They come out of the nucleus now, and they begin to move. And these spindle fibers, which are present in the cytoplasm of the cell, okay. Now, these spindle fibers, they start to assemble. They are preparing the spindle fibers so that now the nucleus divides itself and it can act as a axis for them, or you can say in order to hold on to these chromosomes which are going to divide now. Then the duplicated chromosomes, they condense, and homologous chromosomes, they begin to para. Now, what do you mean by the duplicated chromosomes they condense? What are duplicated chromosomes?
The homologous chromosomes.
Sorry?
The homologous chromosomes.
Yeah, we are saying the homologous chromosomes, they begin to para. But what about the duplicated chromosomes that condense? The duplicated chromosomes are the sister chromatids which are present on each chromosome, isn't it? So they do not divide now, they are intact, connected at the centromere on each homologous chromosome. So these are intact, whereas the homologous chromosomes, they begin to pair up, meaning each from the mother and the father, they pair up like this, the ones which are similar to each other, okay? So these are the sequence of the steps which happen in prophase 1. Now moving on to stage 2, which is metaphase 1, the spindle fibers, they align the homologous chromosomes in the center of the cell, known as the cell equator. Aligning meaning holding them in place. And then each side of the equator has chromosomes from both the parents. These homologous chromosomes which paired up in prophase 1 now are aligned like this, each one from each parent. So these 2 you can consider from father and these 2 from the mother. So this alignment and holding in the center of the cell with the help of the spindle fibers happens in metaphase 1. Now moving on to the third stage, which is anaphase 1, the paired homologous chromosomes, they separate from each other. Have you seen? So this separation which has taken place, did it happen at the centromere? How did they break apart here? The sister chromatids are still attached, isn't it? So did you understand what happened over here? The sister chromatids are still attached, meaning these 2 for each chromosome are still attached. So did it divide into this half and this half now, into 2 halves?
No.
Here? Did it divide? Can you see it here?
Yes.
Okay. So they divided into 2 halves, but the sister chromatids are still attached. Each chromosome did not divide itself into each single sister chromatid, but they divided into half and they were pulled apart with the help of these spindle fibers. Can you see like this? So these pairing up of the homologous chromosomes which happened in step 2, now later these paired homologous chromosomes is separated from each other like this into halves, and they moved towards the opposite sides of the cell. But the sister chromatids are still attached at the centromere like this. All right. So finally, in telophase 1, even the cytoplasm is dividing this one cell into 2 over here. And the spindle fibers, they disassemble and the cell undergoes cytokinesis. Now, each pair of chromosomes here is again covered by a nuclear membrane, forming two different daughter cells. Understood? So these two are unique or different from each other because you have a different combination of the chromosomes in each one of these chromosomes. So by the end of the meiosis 1, you can see two cells which are unique or different from each other. Okay. Now moving on to meiosis 2. Now meiosis 2, it divides the sister chromatids in 4 phases again. Now DNA is not replicated between meiosis 1 and meiosis 2 still. Okay, so you can see in Prophase Two what happens. The centrosomes and centrioles, they move to the opposite sides, but it's happening in 2 cells over here, these 2 daughter cells which were produced in meiosis 1. And the spindle fibers, they start to assemble exactly how it happened in Prophase 1. Then in Metaphase Two, the spindle fibers align the chromosomes along the cell equator like this. And then in Anaphase 2, the sister chromatids are pulled apart. Now the sister chromatids are separated from each other. For each chromosome over here, the sister chromatids are separated and they are pulled apart. Do you see? So this division over here which happened in these 2 chromosomes has separated the 2 sister chromatids and pulled them to the opposite corners of the cell. So the sister chromatids are pulled apart from each other and moved to the opposite sides of the cell over here. And now each cell gives rise to 2 unique cells again. So finally by the end of Telophase 2, which is meiosis 2, you will get 4 haploid cells. The nuclear membranes, they form again around the chromosomes, and spindle fibers, they break apart, the cell cytoplasm divides, and these 2 give rise to 4 haploid unique cells which have a different combination of chromosomes. Got it, ladies? So in mitosis, we studied that each cell divides into 2 identical daughter cells which are exactly the same like the mother cell, whereas in meiosis, by the end of the meiosis 2, it gives rise to 4 unique haploid cells which are very different from each other. Got it, ladies? Any doubts or questions from meiosis 1 and 2? Please ask me if you have any doubts or questions. Is it clear?
Yes.
Leen, Layan, Lana Musa,
Yes, yes.
Dana, Haneen, Habiba. Is it clear, Shuk? Okay, Shuk also says it's clear. All right. So let me also, yeah, you have the same pictures in your textbook as well, ladies, about the meiosis 1 and the meiosis 2. Okay, cytokinesis is the division of the cytoplasm is known as cytokinesis, whereas in meiosis 1 and 2, this division of the nucleus takes place. Did we watch this video yesterday, ladies? The link which we have over here, the phases of meiosis. Did we watch it? Leen?
Which video?
This one from the book over here, the phases of meiosis. Let's watch it. Give me a moment while it's loading. Okay, it's not a video, it's an activity. Study the differences between mitosis and meiosis, then take closer look at the phases of meiosis. Next, explore the concepts of crossing over and independent assortment. So let's start.
Meiosis is a special form of cell division that starts with a diploid cell that has 2 sets of chromosomes, 2n, and results in 4 haploid cells, each having 1 set of chromosomes, n. During meiosis, the number of chromosomes decreases to half the original number by 2 divisions of the nucleus. Meiosis produces the haploid nuclei of gametes from a diploid parent cell nucleus. Organisms in all of the eukaryotic kingdoms use the process of meiosis for sexual reproduction. Sexual reproduction involves the union of gametes from 2 parent organisms. Offspring obtain 1 set of chromosomes from each parent. Variations among offspring arise from chance events and random recombination of chromosomes during meiosis and fertilization.
Did you understand so far? Gamete from parent 1 and parent from parent 2. After the fertilization takes place in the zygote, you can again see 2N because there is 1N from the mother and 1N from the father.
You already know how mitosis divides a parent cell into two identical daughter cells. Meiosis is similar to mitosis in many ways. Some of the phases are similar, and the changes in cell structure are similar. The chromosomes are replicated and divided up. However, meiosis differs from mitosis in the kinds of cells that are produced. Mitosis results in two identical cells, whereas meiosis results in 4 unique cells. Mitosis produces diploid body cells, whereas meiosis produces special haploid cells called gametes. Each gamete that results from meiosis has only half of the parent's chromosomes.
Got it, ladies? Any doubts, questions so far?
The animations above show a cell and its chromosomes as they move through all of the phases of meiosis. As each stage plays, pay attention to the movement of the chromosomes. Also observe how many sister chromatids there are at each stage. Use the Next button to move through the stages. You already know how mitosis divides a parent cell into two identical daughter cells. Meiosis is similar to mitosis in many ways. Some of the phases are similar, and the changes in cell structure are similar. The chromosomes are replicated and divided up. However, meiosis differs from mitosis in the kinds of cells that are produced. Mitosis results in two identical cells, whereas meiosis results in 4 unique cells. Mitosis produces diploid body cells, whereas meiosis produces special haploid cells called gametes. Each gamete that results from meiosis has only half of the parent's chromosomes.
All right, ladies. Now when they are showing the various stages which happen in meiosis 1, I want you to tell me.
The animations above show a cell and its chromosomes as they move through all of the phases of meiosis. As each stage plays, pay attention to the movement of the chromosomes. Also observe how many sister chromatids there are at each stage. Use the Next button to move through the stages.
Then you can see the anaphase 1, and finally the telophase, in which even the cytokinesis divides, and you can see the formation of 2 daughter cells over here, but unique. And again, you're moving on to meiosis 2 here, which is prophase 2, in which again the centrosomes and all of them, the nuclear membrane disintegrate and spindle fibers align these, and the sister chromatids are also separated in meiosis 2. And finally, in telophase 2, you can see the formation of 4 unique haploid cells. Clear so far, ladies? Any doubts or questions you have? All clear?
Yes.
Okay. Others? Layan, Lana, Intisar, the other, all the Janas. Is it clear, ladies? Shog. It's very important for you to understand this process, because again, when we are doing this entire topic, we are talking about the process of meiosis and cell regulation, etc. So it is a little complicated till you understand, but once you know about the 4 stages and what happens, it is very easy. Okay, let's.
Each of these cards has the image of one phase of meiosis. Drag the cards into the correct order.
Okay, so let's do this activity now and check how much you've understood. So prophase 1, which of these pictures matches for prophase 1?
The third last count, I think.
This one?
Yes.
Is this prophase 1? Very good. In prophase 1, you can see all the nuclei are double. It's not single. So let's check whether Lean has the right answer. Perfect. What about metaphase 1? Now, what happens next? You can see this nuclear membrane disintegrated, and you can see the spindle fibers are moved to the corners of the cell. Then what happens next in metaphase 1?
I think it's
This one which I'm pointing at right now?
6.
Oh, this one.
No, the one before it.
This one, right?
Yes.
Okay, so here the spindle fibers have aligned to the homologous chromosomes in the cell equator. So this is what is happening in metaphase 1. Perfect. Then I want the other girls to participate as well. Ladies, what happens in anaphase 1 now? Is this the answer? Wherein the homologous chromosomes are divided and pulled to the either side of the cell?
Yes.
So this is what happens in anaphase 1. And finally, in telophase 1, you can see the formation of 2 unique daughter cells. So which one goes into telophase 1? This one or this one or this one? Or any one of these?
The fourth one.
This one?
No.
Which one is it now?
Yes, this one.
This one?
Yes.
Okay, let me check. Perfect. What about cytokinesis? What is cytokinesis, ladies? What do you mean by cytokinesis? Cytokinesis meaning the division of the cytoplasm. So which one of these pictures represents cytokinesis over here?
That means that the one you just put is for cytokinesis, and another one is for telophase 1.
Okay, in that case, this should go into cytokinesis, you mean?
Yes.
No, this is for telophase itself.
The fourth
This
one?
also shows the cell division, the division of cytoplasm. Sorry, Layan?
Number 5.
Number 5, this one? Or this one?
On the right side.
This
the one that you're clicking.
Which
No, the fourth one.
the mouse pointer is over here. This one or this one? The N or 2N? Or N? Or this one N? This N?
This one.
This one? Okay, here you can see the division of the cell cytoplasm here. So let's see if this is the right answer. Bravo, Layan, right answer. So here it shows the division of the cell cytoplasm, which is known as cytokinesis. Now, which picture matches for prophase 2? Prophase 2.
Second one?
This one?
No, this is
This is Metaphase 2, isn't it? Because the sister chromatids are aligned in the center here.
Yes, it's Metaphase 2, the one beside it.
This one,
Yes.
isn't it? Okay, so let me check. Perfect. This is what is happening in Prophase 2 again. You can see the 2 daughter cells now they are preparing itself to divide further. Now, is this Metaphase 2 where my mouse pointer is right now? Or this one?
The second one.
The second one, wherein the spindle fibers, they are aligning the chromosomes at the cell equator. And then is this true for anaphase 2? This one?
Yes. Yes.
Perfect. And finally, in telophase 2, we can see the formation of 4 unique haploid cells. And how does the cytokinesis happen in over here? So you think this cytokinesis should have gone over here and this cytokinesis here, but though both are the cell cytoplasm division itself, but here it shows in the same cell. So let's check if it matches here. Will it not tell us if it is right or wrong? Okay. Have you seen how many we have done wrong? Okay, so incorrect, try again. Which one is wrong? Telophase 2 is wrong. Obviously, the cytokinesis over here is wrong. And prophase 2 is also wrong. Okay, and telophase 1 is also wrong.
Miss?
Yeah.
I think the one we put for cytokinesis 2 is supposed to be in telophase 1.
Yeah.
And then the one we put for prophase 2 is supposed to be in in telophase 2.
This one should have been in telophase 1.
Yes.
Yeah, perfect. And this one should have been the cytokinesis over here in 1. Right? Let's click on show me. Yeah. Yes, as you said, this should be in telophase 1, and this is for cytokinesis. We did this correct, isn't it? This one is prophase 2. This is metaphase 2, anaphase 2, and this is telophase 2. And finally, after the cytokinesis, they are showing the 4 haploid cells over here. And telophase 2, you can see still only the nucleus has divided, but not the cytoplasm. So this is the correct order, ladies. Shall we do it again, or is it clear to you now? Shall we move on?
It's clear.
Okay, now. So these 1, 2, 3, 4, we did it wrong. So this should be for. In telophase 1 itself, we cannot show the division of the 4 daughter cells. We are basically showing it during the cytokinesis, wherein along with the nucleus, even the cytoplasm is dividing, and then we can show the formation of 2 cells over here at the end of meiosis 1 and 4 cells over here at the end of meiosis 2. This is where we went wrong. All right, great. So let's move on now. I hope it's clear. Alhamdulillah, we did this activity.
Meiosis has special features. 2 divisions of the nucleus, Meiosis I and Meiosis II. Pairing of doubled homologous chromosomes forming tetrads at prophase 1. Crossing over during prophase 1 so that genetic material is exchanged by homologous chromosomes. Separation of homologous chromosomes still doubled in anaphase 1. Production of haploid, not diploid cells. You are going to observe chromosomes in a cell as they go through the stages of meiosis. Describe each phase of meiosis. Notice changes in the homologous chromosomes and sister chromatids from phase to phase.
Okay, so we are in prophase 1 right now. Okay, so number of sets of chromosomes per cell, how many are there? 2 or 1?
2.
2. It is 2n. Number of chromatids per chromosome? 1 or 2?
2.
Obviously it is 2. Status of homologous chromosomes? Independent of each other, paired together in tetrad, separating from each other, or independent in different cells? The homologous chromosomes, how are they in prophase 1? They didn't still pair up. So can we say independent of each other or independent in different cells?
This, I think the right one is separated from each other.
Separating from each other? This one?
Yes, I think so.
Okay. So they are not still paired together. So this could be, okay. So Lean says it's separating from each other. Anybody else who thinks otherwise? Or shall we go with separating from each other?
Independent of each other.
You want to go with independent of each other?
Yes.
Okay, so let's check out which answer is right. Either it is A or C. Let's check C first. How do we know? Oh, it will tell us when we are going further. Status of the sister chromatids. How are the sister chromatids? Paired together, separating from each other, or none? Obviously they are
Paired.
paired together. The sister chromatids, they are present on each chromosome, they are paired together. Yes, Vidi, you have a question?
Yes, I don't think it's separating from each other because I thought this was meiosis 1 in general, but it's only prophase 1.
Only
Right.
Prophase 1. Yeah, that's what I was emphasizing. So you want to go with Lian, independent of each other?
Yes, you can go with Lan's answer. I thought this was the entire process, not only.
No. It's only Prophase 1, wherein still the pairing together of homologous chromosomes did not happen. It will happen in the next phase, which is Metaphase 1. Okay, so let's click independent of each other. Now let's check if our answers are correct. Bismillah. Oops, this is the wrong answer, independent of each other. Then what is the right answer? Show me. Paired together in tetrad. Okay, so though they are paired together, but what is paired together in a tetrad? Meaning there are 4 chromosomes together. Meaning they are paired together in pairs of 2. That is known as a tetrad together. I think so. So let's move on to the next phase now. We have 4 minutes to go. Okay, now this is Metaphase 1. Now let's check out the answers for Metaphase 1. Number of sets of chromosomes per cell. 2 or 1? Ladies?
2.
2 obviously, because still there are 2 sets of chromosomes. Number of chromatids per chromosome.
2.
Again 2. Very good. Status of homologous chromosomes. Independent of each other, paired together in a tetrad, separating from each other or independent in different cells. Now if you could see the picture, what do you think is the status of the homologous chromosomes? They are aligned in the center and held together by the spindle fibers. So can we say they are still paired together in a tetrad? No, they are not, isn't it? So can we say they are separating from each other now?
I don't think so, because they separate in anaphase, not in...
In the next phase, you think they are separating?
Yes.
Alright, so what would be the answer for this phase now? Independent of each other or independent in different cells?
They're not in different cells, they're in the same cells, so maybe it's the first.
So maybe independent of each other? Let's check inshallah. Then status of sister chromatids. Are they still paired together or separating from each other or none?
Right.
They are still paired together, isn't it?
Yes.
Okay. So let's check out. Again, this is the wrong answer, independent of each other. So it is still they are paired together in a tetrad. But the nuclear membrane has disintegrated over here. We have to check out what is the meaning of paired together in a tetrad. I'll check and get back to you, ladies. So basically the answer... Yeah. What is it, Naya?
It's the pair of 2 homologous chromosomes found during Prophase 1 and Meiosis 1.
Could you repeat again?
It's the pair of 2 homologous chromosomes found during Prophase 1 and Meiosis 1.
Okay, so still they are in pair itself, so they are not separated. So the pair of homologous chromosomes in Prophase 1, in Meiosis 1, they are known as a tetrad. So still in the second phase, which is Metaphase 1, also they are paired together because they are still paired but aligned at the center. So in the next stage, they will be separated. So that is why still we are saying paired together in a tetrad. 1 minute to go. Okay. Shall we quickly do this? Anaphase 1. Number of sets of chromosomes per cell. How many do we have now?
2.
Number of chromatids per chromosome.
2.
Status of homologous chromosomes.
Separating from each other.
Separating from each other. Status of sister chromatids.
They're still paired.
Paired together. Perfect. We'll do the next activity in our next class, inshallah. It's 11:10. Meanwhile, if you want to do it, I can send the link, but it doesn't work without the username and password. So let me check as to how I can add you all to this class so that you can have access to all these interesting activities, inshallah. Regarding the test marks, ladies, so please check out if you have any doubts or questions, you can come in contact with me. I will give you the marks. Sure, very good. All right, 37 attendees. Thank you so much, ladies. Take very good care of yourselves and have a wonderful weekend ahead. See you again next week.